In 1982, a historical note on a series of evolutionary biology books, F, could state without qualification that evolution is the central organizing principle of biology. Smokovitis commented on this that, what the architects of the synthesis had worked to construct had by 1982 become a matter of fact, adding in a footnote that, the centrality of evolution had thus been rendered tacit knowledge, part of the received wisdom of the profession. In 1982, a historical note on a series of evolutionary biology books, F, could state without qualification that evolution is the central organizing principle of biology. Smokovitis commented on this that, what the architects of the synthesis had worked to construct had by 1982 become a matter of fact, adding in a footnote that, the centrality of evolution had thus been rendered tacit knowledge, part of the received wisdom of the profession. 95. By the late 20th century, however, the modern synthesis was showing its age, and fresh syntheses to remedy its defects and fill in its gaps were proposed from different directions. These have included such diverse fields as the study of society, 87 developmental biology, 50 epigenetics, 96 molecular biology, microbiology, genomics, 3 symbiogenesis, and horizontal gene transfer. 97. The physiologist Dennis Noble argues that these additions render neo-Darwinism in the sense of the early 20th century's modern synthesis, at the least, incomplete as a theory of evolution. 97. And one that has been falsified by later biological research. 97. Michael Rose and Todd Oakley note that evolutionary biology, formerly divided and balkanized, has been brought together by genomics. It has in their view discarded at least 5 common assumptions from the modern synthesis, namely that the genome is always a well-organized set of genes, that each gene has a single function, that species are well adapted biochemically to their ecological niches, that species are the durable units of evolution, and all levels from organism to organ, cell and molecule within the species are characteristic of it, and that the design of every organism and cell is efficient. They argue that the new biology integrates genomics, bioinformatics, and evolutionary genetics into a general-purpose toolkit for a postmodern synthesis. 54. Pigliucci's Extended Evolutionary Synthesis, 2007. In 2007, more than half a century after the modern synthesis, Massimo Pigliucci called for an extended evolutionary synthesis to incorporate aspects of biology that had not been included or had not existed in the mid-20th century. 98-99. It revisits the relative importance of different factors, challenges assumptions made in the modern synthesis, and adds new factors. 99-100, such as multilevel selection, transgenerational epigenetic inheritance, niche construction, and evolvability. 101. 96-102. Kunin's Postmodern Evolutionary Synthesis, 2009. A 21st century tree of life showing horizontal gene transfers among prokaryotes and the saltational endosymbiosis events that created the eukaryotes, neither fitting into the 20th century's modern synthesis. In 2009, Darwin's 200th anniversary, The Origin of Species 150th, and the 200th of Lamarck's Early Evolutionary Synthesis. 3. Philosophie zoologique. The evolutionary biologist Eugene Koonin stated that while the edifice of the early 20th century modern synthesis has crumbled, apparently, beyond repair, 3. A new 21st century synthesis could be glimpsed. 3 interlocking revolutions had, he argued, taken place in evolutionary biology, molecular, microbiological, and genomic. The molecular revolution included the neutral theory, that most mutations are neutral and that negative selection happens more often than the positive form, and that all current life evolved from a single common ancestor. In microbiology, the synthesis has expanded to cover the prokaryotes, using ribosomal RNA to form a tree of life. Finally, genomics brought together the molecular and microbiological syntheses in particular. Horizontal gene transfer between bacteria shows that prokaryotes can freely share genes. Many of these points had already been made by other researchers such as Ulrich Kitzkera and Carl J. Nicholas. 103. Towards a replacement synthesis. Inputs to the modern synthesis, with other topics, inverted colors, such as developmental biology that were not joined with evolutionary biology until the turn of the 21st century. 103. Biologists, alongside scholars of the history and philosophy of biology, have continued to debate the need for, and possible nature of, a replacement synthesis. For example, in 2017 Philippe Huneman and Dennis M. Walsh stated in their book Challenging the Modern Synthesis that numerous theorists had pointed out that the disciplines of embryological developmental theory, morphology, and ecology had been omitted. They noted that all such arguments amounted to a continuing desire to replace the modern synthesis with one that united all biological fields of research related to evolution, adaptation, and diversity in a single theoretical framework. 104. They observed further that there are 2 groups of challenges to the way the modern synthesis viewed inheritance. The first is that other modes such as epigenetic inheritance, phenotypic plasticity, the Baldwin effect, and the maternal effect allow new characteristics to arise and be passed on and for the genes to catch up with the new adaptations later. The second is that all such mechanisms are part, not of an inheritance system, but a developmental system. The fundamental unit is not a discrete selfishly competing gene, but a collaborating system that works at all levels from genes and cells to organisms and cultures to guide evolution. 105. The molecular biologist Sean B. Carroll has commented that had Huxley had access to evolutionary developmental biology, embryology would have been a cornerstone of his modern synthesis, and so Evo-Devo is today a key element of a more complete, expanded evolutionary synthesis. 106. Historiography. Looking back at the conflicting accounts of the modern synthesis, the historian Betty Smokovitis notes in her 1996 book Unifying Biology, the evolutionary synthesis and evolutionary biology that both historians and philosophers of biology have attempted to grasp its scientific meaning, but have found it a moving target. 107. The only thing they agreed on was that it was a historical event. 107. In her words. By the late 1980s, the notoriety of the evolutionary synthesis was recognized. So notorious did the synthesis become that few serious historically minded analysts would touch the subject, let alone know where to begin to sort through the interpretive mess left behind by the numerous critics and commentators. Genetic variation. Genetic variation is the difference in DNA among individuals, 1. Or the differences between populations among the same species. 2. The multiple sources of genetic variation include mutation and genetic recombination. 3. Mutations are the ultimate sources of genetic variation, but other mechanisms, such as genetic drift, contribute to it, as well. 2. Darwin's finches or Galapagos finches. 4. Parents have similar gene coding in this specific situation where they reproduce and variation in the offspring is seen. Offspring containing the variation also reproduce and passes down traits to their offspring. Among individuals within a population, genetic variation can be identified at many levels. Identifying genetic variation is possible from observations of phenotypic variation in either quantitative traits that vary continuously and are coded for by many genes, e.g., leg length in dogs, or discrete traits that fall into discrete categories and are coded for by one or a few genes, e.g., white, pink, or red petal color in certain flowers. Genetic variation can also be identified by examining variation at the level of enzymes using the process of protein electrophoresis. 5 polymorphic genes have more than one allele at each locus. Half of the genes that code for enzymes in insects and plants may be polymorphic, whereas polymorphisms are less common among vertebrates. Ultimately, genetic variation is caused by variation in the order of bases in the nucleotides in genes. New technology now allows scientists to directly sequence DNA, which has identified even more genetic variation than was previously detected by protein electrophoresis. Examination of DNA has shown genetic variation in both coding regions and in the non-coding intron region of genes. Genetic variation will result in phenotypic variation if variation in the order of nucleotides in the DNA sequence results in a difference in the order of amino acids in proteins coded by that DNA sequence, and if the resultant differences in amino acid sequence influence the shape, and thus the function of the enzyme. 6. Between populations. Differences between populations resulting from geographic separation is known as geographic variation. Natural selection, genetic drift, and gene flow can all contribute to geographic variation. 7. Measurement. Genetic variation within a population is commonly measured as the percentage of polymorphic gene loci or the percentage of gene loci in heterozygous individuals. The results can be very useful in understanding the process of adaptation to the environment of each individual in the population. 8. Sources. A range of variability in the Musseldonax variabilis. Random mutations are the ultimate source of genetic variation. Mutations are likely to be rare, and most mutations are neutral or deleterious, but in some instances, the new alleles can be favored by natural selection. Polyploidy is an example of chromosomal mutation. Polyploidy is a condition wherein organisms have 3 or more sets of genetic variation, 3n or more. Crossing over, genetic recombination, and random segregation during meiosis can result in the production of new alleles or new combinations of alleles. Furthermore, random fertilization also contributes to variation. Variation and recombination can be facilitated by transposable genetic elements, endogenous retroviruses, lines, signs, etc. For a given genome of a multicellular organism, genetic variation may be acquired in somatic cells or inherited through the germline. Forms. Genetic variation can be divided into different forms according to the size and type of genomic variation underpinning genetic change. Small-scale sequence variation, less than 1 kilobase, kb, includes base pair substitution and indels. 9. Large-scale structural variation, greater than 1 kilobit, can be either copy number variation, loss or gain, or chromosomal rearrangement translocation, inversion, or segmental acquired unapparental disomy. 9. Genetic variation and recombination by transposable elements and endogenous retroviruses sometimes is supplemented by a variety of persistent viruses and their defectives which generate genetic novelty in host genomes. Numerical variation in whole chromosomes or genomes can be either polyploidy or aneuploidy. Maintenance in populations. A variety of factors maintain genetic variation in populations. Potentially harmful recessive alleles can be hidden from selection in the heterozygous individuals. In populations of diploid organisms, recessive alleles are only expressed in the less common homozygous individuals. Natural selection can also maintain genetic variation in balanced polymorphisms. Balanced polymorphisms may occur when heterozygotes are favored or when selection is frequency dependent. RNA viruses. A high mutation rate caused by the lack of a proofreading mechanism appears to be a major source of the genetic variation that contributes to RNA virus evolution. 10. Genetic recombination also has been shown to play a key role in generating the genetic variation that underlies RNA virus evolution. 10. Numerous RNA viruses are capable of genetic recombination when at least 2 viral genomes are present in the same host cell. 11. RNA recombination appears to be a major driving force in determining genome architecture in the course of viral evolution among Picornaviridae plus ssRNA, e.g. poliovirus. 12. In the Retroviridae plus ssRNA, e.g. HIV. Damage in the RNA genome appears to be avoided during reverse transcription by strand switching, a form of genetic recombination. 13, 14, 15. Recombination also occurs in the coronaviridae plus ssRNA, e.g. SARS. 16. Recombination in RNA viruses appears to be an adaptation for coping with genome damage. 11. Recombination can occur infrequently between animal viruses of the same species but of divergent lineages. The resulting recombinant viruses may sometimes cause an outbreak of infection in humans. 16. History of genetic variation. Evolutionary biologists are often concerned with genetic variation, a term which in modern times has come to refer to differences in DNA sequences among individuals. However, quantifying and understanding genetic variation has been a central aim of those interested in understanding the varied life on Earth since long before the sequencing of the first full genome, and even before the discovery of DNA as the molecule responsible for heredity. While today's definition of genetic variation relies on contemporary molecular genetics, the idea of heritable variation was of central importance to those interested in the substance and development of life even before the writings of Charles Darwin. The concept of heritable variation, the presence of innate differences between life forms that are passed from parents to offspring, especially within categories such as species, does not rely on modern ideas of genetics, which were unavailable to 18th and 19th century minds. Pre-Darwinian concepts of heritable variation. In the mid-1700s, Pierre-Louis Maupertuis, a French scholar now known primarily for his work in mathematics and physics, posited that while species have a true, original form, accidents during the development of nascent offspring could introduce variations that could accumulate over time. 17. In his 1750 Essai de Cosmologie, he proposed that the species we see today are only a small fraction of the many variations produced by a blind destiny, and that many of these variations did not conform to their needs, thus did not survive. 18. In fact, some historians even suggest that his ideas anticipated the laws of inheritance further developed by Gregor Mendel. 19. Simultaneously, French philosopher Denis Diderot proposed a different framework for the generation of heritable variation. Diderot borrowed Maupertuis's idea that variation could be introducced during reproduction and the subsequent growth of offspring. 20. And thought that production of a normal organism was no more probable than production of a monstrous one. 21. However, Diderot also believed that matter itself had lifelike properties and could self-assemble into structures with the potential for life. 20. Diderot's ideas on biological transformation, introduced in his 1749 work Letter on the Blind, were thus focused on variability of spontaneously generated forms, not variability within existing species. 22. Both Maupertuis and Diderot built on the ideas of Roman poet and philosopher Lucretius, who wrote in De Rerum Natura that all the universe was created by random chance, and only the beings that were not self-contradictory survived. 23. Maupertuis's work is distinguished from the work of both Lucretius and Diderot in his use of the concept of conformity in explaining differential survival of beings, a new idea among those who believe that life changed over time. 23. Like Diderot, two other influential minds of the 18th century, Erasmus Darwin and Jean-Baptiste Lamarck, believed that only very simple organisms could be generated by spontaneous generation, so another mechanism was necessary to generate the great variability of complex life observed on Earth. 17. Erasmus Darwin proposed that changes acquired during an animal's life could be passed to its offspring, and that these changes seemed to be produced by the animal's endeavors to meet its basic needs. 24. Similarly, Lamarck's theory of the variability among living things was rooted in patterns of use and disuse, which he believed led to heritable physiological changes. 17. Both Erasmus Darwin and Lamarck believed that variation, whether it arose during development or during the animal's life, was heritable, a key step in theories of change over time extending from individuals to populations. In the subsequent century, William Herschel's telescopic observations of diverse nebulae across the night sky suggested to him that different nebulae could each be in different stages in the process of condensation. This idea, which came to be known as the nebular hypothesis, suggested that natural processes could both create order out of matter and introduce variation, and that these processes could be observed over time. 17. While it may seem to the modern reader that astronomical theories are irrelevant to theories of organic variation, these ideas became significantly conflated with ideas of biological transformation, what we now know as evolution, in the mid-19th century, laying important groundwork for the work of subsequent thinkers such as Charles Darwin. 25. Darwin's concept of heritable variation. Charles Darwin's ideas of heritable variation were shaped by both his own scientific work and the ideas of his contemporaries and predecessors. 26. Darwin ascribed heritable variation to many factors, but particularly emphasized environmental forces acting on the body. His theory of inheritance was rooted in the now disproven idea of gemmules, small, hypothetical particles, which capture the essence of an organism and travel from all over the body to the reproductive organs, from which they are passed to offspring. 27. Darwin believed that the causal relationship between the environment and the body was so complex that the variation this relationship produced was inherently unpredictable. 28. However, like Lamarck, he acknowledged that variability could also be introduced by patterns of use and disuse of organs. 29. Darwin was fascinated by variation in both natural and domesticated populations, and his realization that individuals in a population exhibited seemingly purposeless variation was largely driven by his experiences working with animal breeders. 30. Darwin believed that species changed gradually, through the accumulation of small, continuous variations, a concept that would remain hotly contested into the 20th century. 31. Post-Darwinian concepts of heritable variation. In the 20th century, a field that came to be known as population genetics developed. This field seeks to understand and quantify genetic variation. 31. The section below consists of a timeline of selected developments in population genetics, with a focus on methods for quantifying genetic variation. 1866 Heterozygosity. Gregor Mendel's hybridization experiments introduced the concept that in the 1950s came to be recognized as heterozygosity. 29. In a diploid species, one that contains 2 copies of DNA within each cell, one from each parent, an individual is said to be a heterozygote at a particular location in the genome if its 2 copies of DNA differ at that site. Heterozygosity, the average frequency of heterozygotes in a population, became a fundamental measure of the genetic variation in a population by the mid-20th century. 32. If the heterozygosity of a population is 0, every individual is homozygous, that is, every individual has 2 copies of the same allele at the locus of interest and no genetic variation exists. 1918 Variance. In a seminal paper entitled, The Correlation Between Relatives on the Supposition of Mendelian Inheritance, R.A. Fisher introduced the statistical concept of variance, the average of squared deviations of a collection of observations from their mean. Whereas the variance n is the mean of the population from which the observations are drawn. 33. R. Fisher's work in population genetics was not just important to population genetics, these ideas would also form the foundations of modern statistics. 1918, 1921 Additive and dominant genetic variance. R. A. Fisher subsequently subdivided his general definition of variance into 2 components relevant to population genetics, additive and dominant genetic variance. 34. An additive genetic model assumes that genes do not interact if the number of the genes affecting the phenotype is small and that a trait value can be estimated simply by summing the effect of each gene on the trait. Under Fisher's model, the total genetic variance is the sum of the additive genetic variance, the variance in a trait due to these additive effects, and the dominant genetic variance which accounts for interactions between genes. 33. 1948 Entropy. Unlike variance, which was developed with the purpose of quantifying genetic variance, Claude Shannon's measure of diversity, now known as Shannon entropy, was developed as part of his work in communication theory as a way to quantify the amount of information contained in a message. However, the method quickly found use in population genetics, and was the central method used to quantify genetic diversity in a seminal paper by Richard Lewontin, The Apportionment of Human Genetic Diversity. 35. 1951 F statistics. F statistics, also known as fixation indices, were developed by population geneticist Sewell Wright to quantify differences in genetic variation within and between populations. The most common of these statistics, FST, considers in its simplest definition 2 different versions of a gene, or alleles, and 2 populations that contain one or both of these 2 alleles. FST quantifies the genetic variability among these 2 populations by computing the average frequency of heterozygotes across the 2 populations relative to the frequency of heterozygotes if the 2 populations were pooled. 36. F statistics introduced the idea of quantifying hierarchical concepts of variance and would become the foundation of many important population genetic methods, including a set of methods that tests for evidence of natural selection in the genome. 37. Genetic variation. Genetic variation is the difference in DNA among individuals, 1, or the differences between populations among the same species. 2. The multiple sources of genetic variation include mutation and genetic recombination. 3. Mutations are the ultimate sources of genetic variation, but other mechanisms, such as genetic drift, contribute to it, as well. 2. Darwin's finches are Galapagos finches. 4. Parents have similar gene coding in this specific situation where they reproduce and variation in the offspring is seen. Offspring containing the variation also reproduce and passes down traits to their offspring. Among individuals within a population, genetic variation can be identified at many levels. Identifying genetic variation is possible from observations of phenotypic variation in either quantitative traits, traits that vary continuously and are coded for by many genes, e.g., leg length in dogs, or discrete traits, traits that fall into discrete categories and are coded for by one or a few genes, e.g., white, pink, or red petal color in certain flowers. Genetic variation can also be identified by examining variation at the level of enzymes using the process of protein electrophoresis.